Thanks everyone joining online. Yeah, I'd like to introduce Greg Radick, who's a professor of history and philosophy of science at the University of Leeds, who's adding to our set of Mendel seminars on disputed inheritance, the battle over Mendel, and the future of biology. Great.
Thank you, George. Thanks everybody who's come. Thanks everyone online. It's a real honor to be joining this birthday bash for Gregor Mendel. A little bit late, but that doesn't matter, right? Because Mendel is worth celebrating, and he's worth celebrating for an achievement which is rather wittily encoded in this anniversary image, right? Of yellow seeds and yellow peas and green peas, and we see the yellow to green in a ratio of 3 to 1. So Mendel discovers this pattern in crossing his peas in the garden monastery in Brunn, and he explains it brilliantly, right? On the supposition that the hybrid plants, when they produce gametes, the gametes are not themselves hybrid, but are pure for one or the other of the parental characters. And it goes on to become foundational for the science that we call genetics. And everybody knows this, because whenever you study genetics at any level, you're going to meet Mendel and his peas. You don't even need to go to school to encounter Mendel and his peas. This is from my children's Horrible Science Annual. It's a good book. The Horrible Science is the name of the series. And we see it gives a very good account of Mendelian genetics, but also makes some good gags. Here we have Mendel labeling out pea soup in the monastery canteen, but explaining to one that there's no peas for the wicked, so no soup for him. The anniversary year saw some good gags. What did Gregor Mendel say when he founded genetics? Those in the room, all these PhDs, what do you think he said? Whoopee! Right. And so some good gags in the anniversary year, a lot of love for Gregor Mendel, and along with a lot of love and some gags, there was a certain number of pretty familiar propositions about Mendel. I'm going to highlight 3 of them. The first is that he discovered the gene, understood as the atom of heredity. Second proposition, that our present-day knowledge of genes, right, what we know about them and what we know how to do with them and even to them, we owe to that discovery. And I'm going to round it out with a third proposition, that enlightened understanding of biological inheritance, right? Getting serious about it, getting scientific in a way that matters in the history of science, as in the science classroom, starts with Mendel. I'm going to call all of that the received wisdom on Mendel. And in calling it that, it won't surprise you to learn that I'm here to question it. I'm here to probe it, to test it, to be a little bit skeptical. And toward the end, we'll talk a little bit together about where that leaves Mendel in our thinking and in our teaching. Let me start with that first item of received wisdom, that Mendel discovered the gene, right, the atom of heredity. I'm going to talk least about this one because I think it's probably a topic that's come up quite a lot in the Bristol seminar already. In my view, which isn't in any way original to me, here I'm very largely following what I take to be the good stuff coming out of scholarly history of science. The key to getting straight on what Mendel's paper is about is to take his title seriously. Experiments on not heredity, right, but on plant hybrids. And Mendel explains in the paper that in his view, there are 2 kinds of hybrids. One kind is what he calls the constant hybrid. So you've got plant with interesting character A, plant with interesting character B. You cross them together, you get plant with interesting character C. Then you cross your fingers, let that plant self-fertilize. And if you're lucky, interesting character C carries on. That's what Mendel calls a constant hybrid. Mendel says, that's not what my paper's about. My paper is about the other class of hybrids, right? Where you've got plant with interesting character A, plant with interesting character B, you cross them together, you get plant with interesting character C, you cross your fingers and you let them self-fertilize and it doesn't work out. The old character returns. Mendel calls these the variable hybrids. Mendel says, this is what my paper's about. this class of hybrids. I want to know, says Mendel, whether there is a law that governs the return of the vanished character. And if so, what is that law? And furthermore, if I can identify the law, can I explain it? And boy, does he do that. Absolutely brilliantly. But that's what the paper is about. It's not about heredity. The word heredity comes up. A form of it comes up in passing at one moment in the paper. It's about what he says it's about. It's about hybrids. And I think an interesting kind of empirical test for this way of interpreting Mendel's ambitions in the paper is to check out Mendel's annotations on his copy of Darwin's On the Origin of Species. Inheritance mattered a lot to Charles Darwin under that heading. And in chapter 1 of The Origin, he has an explicitly labeled discussion about inheritance. How many annotations do you think Mendel made on Darwin's passages on inheritance? 0. Almost all of his annotations on his copy of The Origin of Species are on the chapter that a lot of us will spend a lot of time with, the chapter on hybrids. There's a little bit more to be said about that, but I think that's an interesting empirical test for asking what was on Mendel's mind in the early 1860s as he's finishing up his experiments, beginning to think about writing his paper. So it's a paper about hybrids, like it says. And if we ask ourselves, well, what about that notion of the atom of heredity, an atomic conception, right, in Mendel's explanation of his patterns, right? We sort of can't help but read the paper, assuming that Mendel, like us, thought of whatever it was that underpinned those visible characters as countable somethings, right? Atomic in that sense. But as others have pointed out, it's entirely consistent with everything that Mendel writes about in the paper, that he didn't have an atomic conception of the matter underpinning the visible characters. You can read the paper and suppose that instead he thought of them as something like fluids, immiscible fluids like oil and water. Or if we go to the elementary Mendelian pattern, right, and think about what happens to the gametes that come together to form the first generation of hybrids on a kind of fluid reading, the yellowness making stuff and the greenness making stuff come together. They don't fuse. They stay together in a kind of unstable union. And then when gametes are produced, that union dissolves. There's almost a kind of contents under pressure release. And the yellowness goes into its own gametes, and the greenness making stuff goes into its own gametes. So the notion of Mendel, the author of the atom of heredity notion, is as problematic as the notion that Mendel's paper is devoted to heredity. So let me give you some interim take-homes from this part of my skeptical probing of the received wisdom on Mendel. The first, to put it in a bumper sticker, is that Mendel was no Mendelian. This is a famous phrase. I'm from the University of Leeds. Valuably was at the University of Leeds when he published that paper. The slogan's actually due to his colleague, my colleague, John Hodge, and it's become weirdly irritating to a certain class of geneticists. But I think surely if we want to celebrate Mendel in his bicentenary year, we want to celebrate the reality of Mendel, not some fictionalized version of Mendel. And a little reflection, I think, ought to lead us to see that it's far more likely that Mendel had his questions and his thoughts rather than our questions and our thoughts. That's what the slogan is meant to gesture towards. Second take home for now, the reason that he concentrated at the start on either or uniform characters, right? Greenness or yellowness, roundedness or wrinkledness, caused by nothing other than associated cellular material, was because he was interested in tracking the fate of hybrid characters. This is why that matters, right? What was Mendel's question? When you see what his question was, you can see why his methodological choices make a lot of sense. They're brilliant for those purposes. By contrast, those same choices construed as starting points for understanding inheritance have been and remain very problematic. And I'll be coming back to that point. So that's all I wanted to say on this first item of received wisdom about Mendel, right? The discoverer of the gene understood as the atom of heredity. Let me come on to my second item, that our present day knowledge of genes, what we know about them and what we know how to do with them and even to them, we owe to that discovery. The idea of continuity between what happened in Mendel's monastery garden in the 1850s and 60s and what has happened since, particularly in the 20th century, with the molecularization of heredity, is a familiar theme in writing about Mendel, particularly when it's anniversary time. And here it's represented iconographically, right? The notion that the P seeds and the double helix, they belong together, right? It's one amalgam, one story. Well, I think one good way to begin being a little bit more independently minded about that notion is to think a little bit about chromosomes and inheritance. Typically, our textbooks say, oh, well, chromosomes only really become part of the story with T.H. Morgan and his students and the work they did, brilliant work on fruit flies, beginning in about 1910. I'm going to tell you a different story. I'm going to begin my version for today anyway, in March 1900. That's a big year for Mendelian-minded folks because it's in March 1900 that Hugo de Vries' paper on Mendelian ratios and Mendelian explanations is read at the Académie des Sciences in Paris. In that very week, J. Arthur Thomson, who was a kind of Brian Cox of his day, he is the well-known, popular, really smart, classy science popularizer, gives a lecture at the Royal Institution in London on the facts of inheritance. That's his title. And among the facts of inheritance that he tells ordinary people who show up at the Royal Institution on a Friday evening to hear about what's up at the science frontier, he talks about the cellular basis of inheritance. And I'll just pick out a few highlights of what he has to say. He says that within the fertilized egg, remember, this is March 1900, right? Within the fertilized egg, the true bearers of hereditary qualities are mainly, but not exclusively, in the cell nucleus. Within the nucleus, they're probably in the rod-shaped chromosomes, right? Just remind yourself, are literally the colored bodies because they became visible under the microscope with certain stains. Now, what's in there? What's in those chromosomes that could make them enjoy this status within a body? Thompson's really smart. He says, well, plainly not complete determinations of inherited characters. That can't be what's in the chromosomes. So what is in there? He has a nice phrase. He calls them initiatives. Initiatives that start the chains of cause and effect, which in the course of development, within the right kind of environment, culminate in visible characters. So that's popular science. It's classy popular science, but it's popular science in March 1900. Now, I only have time today to kind of give you a whistle-stop tour of what happens to chromosomes in scientific understanding between then and now. But a few stops on the whistle-stop tour are worth mentioning. By the 1940s, right, it becomes clear that genes direct the formation of enzymes, more generally of proteins. So biochemically, the mechanism that seems to relate a bit of chromosome to a body is becoming clearer. By the 1950s, the smart money starts to be on DNA as the biomolecule that matters in chromosomes. And of course, in 1953, Watson and Crick work out the double helical structure at a stroke, making sense of this extraordinary property that genes have of being able both to replicate and to direct the synthesis of proteins. By the end of the 1960s, the genetic code, as it comes to be known, is worked out. Stunning work relating sequences of nucleotides in the DNA to sequences of amino acids in the proteins. In the 1970s, for the first time, the ability to do things with DNA, to sequence strands of DNA, but also to cut bits out and paste them in other places, genetic engineering as it's known at the time. Now, if we stand back from my highlights, but also the larger history of the growth of knowledge, the chromosomal molecular basis of inheritance, and we ask ourselves about the relationship between all of that and Mendelian genetics, I think we have, broadly speaking, two options. One is to suppose that this chromosomal molecular knowledge of genes just deepened the prior Mendelian knowledge of genes by increasing the level of resolution. So you could think about genetic knowledge as a kind of magnifying glass. And over the course of the 20th century, through intellectual progress, through technological progress, the power of magnification amped up. So what you got is the same knowledge at deeper and deeper levels. That's one way of thinking about what happened in the course of the 20th century. But there's another way, which I find more appealing and true. And that is that actually this new knowledge, in effect, replaced Mendelian knowledge. It wasn't the same knowledge, only deeper. Molecular knowledge comes out of different sciences, right? It comes out of X-ray crystallography, biochemistry, done by different people. And what we end up having is a union, if you want to describe it like that, but in other ways, a very heterogeneous body of knowledge, which fits together in complex ways. By way of giving yourself a way into that latter thought, consider how often dominant and recessive versions have bottomed out in differences in the quantity expressed of the same protein, right? With the character itself turning out to be spectral in form and multifactorial in its causes. Let me give you two examples to think with. First of all, eye color, right? Who hasn't heard that brownness is dominant in human eye color, blueness is recessive, therefore two blue-eyed parents can't have a brown-eyed child. And one is led to believe that in the eye there must be some brownness pigment, which is pumped out by the brownness allele, and some blueness pigment, which is pumped out by the blueness allele. And in a heterozygote, the brownness somehow overmasters the fragile blueness from the blueness allele. But what one learns when one learns the biochemical story is that there's nothing like that. There's just melanin. And depending on the quantity of melanin and a lot of other complexities, but fundamentally the quantity of melanin, you can get eyes which are every which color, between very, very blue and very, very brown. And we all, of course, know that. And yet the fiction of two character states, brownness or blueness, simply dominant or simply recessive, carries on to the point where the experts in eye color genetics beg teachers, please stop teaching students that eye color is a simple Mendelian character. It just isn't. It's polygenic in its causation, and it's spectral in its phenotype. Another character a little closer to home for the Mendel anniversary year, Mendel's peas, right? Yellowness and greenness. And again, one's invited to think, well, there must be kind of yellowness being pumped out by the yellowness making factors and greenness being pumped out by the greenness making factor. And in the heterozygote, yellowness overmasters greenness. And it's only when greenness is free from the overmastering effects of yellowness that we get to see greenness. But biochemically, again, it's nothing like that. There is simply enzyme that degrades chlorophyll. And depending on how much of it you can get different degrees of yellowness and greenness. And this was pointed out in 1902. So that's 2 years, right, after the rediscovery of Mendel's paper, 2 years after Mendel's paper begins to be a talking point. This photograph is published. I'll tell you a little bit more about it in just a few minutes, but for the moment, have a look at the very top of the image. And you can see there by number 1, yeah, there are some really green peas. And in number 6, yeah, there are some really yellow peas. But if you're classifying peas that were grown by other breeders, and you're not intent on using Mendel's binary categories, you can identify peas that fall along the color continuum from greenness to yellowness. So it appears that, sum up in an interim way for now, right? Genes understood in chromosomal and molecular terms are not just elementary Mendelian genes at higher resolution, but they're different sorts of entity altogether. And one way of thinking about what that difference might be is to think about them this way, that with elementary, I underscore that, elementary Mendelian conceptions, we're dealing with a character maker, yellowness, greenness. Whereas when we've got a sense of the molecular and biochemical mechanism that stretches between a chromosome and the visible phenotype, we find it far easier to make sense of the contribution of a gene variant as a difference maker, something that makes a difference to a complex causal chain, rather than the maker of a character in and of itself. And arguably, and I go into this kind of thinking much more in the book that George kindly mentioned, we would have achieved such an understanding anyway, once we figured out that chromosomes were the cellular basis of inheritance. And again, that was already taken seriously in 1900. So if we were to rerun the history of genetics, but without Mendelism, which isn't the same thing as without Mendel, but without Mendelism, without Mendelian conceptions and patterns taking over the shop in the way that they did, I venture that we would have learned pretty much what we've learned about the molecular and chromosomal nature of inheritance. And Mendelian crossing would have had its place as a tool, a valuable tool, useful in its place, but not as the big generalization around which to hang everything else when it comes to understanding inherited characters and how they work. And in the book, I bring this point home by way of a little story about something that long fascinated me when I first learned about it, and that there are goats now in Utah which produce spider silk in their milk, transgenic goats. So it turns out that you can, in the first instance, take a spider silk gene out of a spider, put it into a goat, and it works. The actual story is a little more complex than that, but what matters for our purposes is that the doing of that work didn't depend a single jot on Mendelian genetics. It turns out that you can't farm spiders. So the spider silk is interesting industrially, but you can't farm spiders because they just eat each other. So you can't cross them either. The work that was done in the 80s and 90s didn't depend in the slightest on Mendelian knowledge about this, right? What the investigators did was to sequence enough of the silk protein that interested them, work out from that what the RNA looks like, scooped it out of the silk-making gland in a spider, the mRNA transcripts, and then used the DNA library to find eventually the DNA that they needed in order to begin making more of it in goats. Didn't depend, as I said, in the slightest on anything Mendelian. So we've looked at the notion that Mendel discovered the gene, considered as the atom of heredity. We've looked at the idea that our present-day knowledge of genes depends in any kind of heavy way on what Mendel did in the monastery garden in the 1850s and 60s. I now want to turn in my skeptical way to the third item on my list of received wisdom about Mendel. That enlightened understanding of biological inheritance in the history of science, as in the science classroom, starts with Mendel. And here I'm going to take you back to that amazing photograph from 1902. It's in a paper by this gentleman, not very well remembered outside specialist circles. His name is Walter Frank Raphael Weldon, Raphael to his friends. In 1902, he was a professor of comparative anatomy at the University of Oxford. When he began in 1900 to hear that there was a buzz around this Mendel paper, he got interested in it, but he quickly found himself persuaded that there was some problems with this paper, that it wasn't anything like the key to a universal understanding of inheritance. When he published his paper in 1902, as he knew would, he seriously provoked his former friend turned frenemy, William Bateson, who at that point was based at Cambridge University. And from that moment onward, these two were in a battle. And that's actually how they describe it. I hope that my book succeeds in projecting the reader into the thick of an intellectual debate where the stakes were high for the two people who best understood what those stakes were and how important it was. Their debate ended when Weldon unexpectedly and prematurely, at the age of 46, died of pneumonia, spring 1906, with the grand work that he was then in the midst of a book setting out his vision, his alternative vision for the science of inheritance, unfinished and unpublished. So I think this is important enough that I've written a 600-page book about it. It will be out in just a few months. I can only today give you a little sense of what I get up to in the book. But let me carry you forward in Weldon's story from 1902, February 1902, he publishes the article in which that photograph of Pease appears. Later that same year, at the British Association for the Advancement of Science meeting, which happened in Belfast that year in the autumn, he gives a public lecture on inheritance. So where do you start? Where do you start your audience off? Your public audiences come. Where do you begin them in thinking about the state of the art on inheritance in 1902? With Weldon, it's with the water flea Daphnia. So he early on in this lecture discusses an experimental study done by a student of his on the growth of the spine in this water flea. And what he tells his audience is that if you raise Daphnia in overcrowded water, you will produce spineless Daphnia generation after generation, right? The water becomes poisoned with their waste, and that then affects their development. But the moment you remove, right, one of the spineless individuals to pure water, its descendants will grow up to have a long spine. Here's Weldon, a quotation. Now, clearly the condition of the spine is not exclusively acquired, and it is not exclusively inherited. It belongs to both categories. This, by the way, is a mantra for Weldon. He repeats this in various forms over and over and over again. He thinks that biology has got so messed up, largely due to Weismann, for adopting this phrase, acquired character, as if that made any sense at all. Back to Weldon, he says, a Daphnia of certain ancestry, right, we would say genetic background, growing in a solution of certain chemical composition, has a spine of a certain length. You cannot assert that either factor is more essential than the other. For if the Daphnia remain the same, you can change the result by changing the solution. While if the solution remains the same, you can, within considerable limits, change the result by changing the Daphnia. We see here the take home, right? The complex way in which the characters of an organism are determined by the interaction of the two sets of factors, the environmental and the ancestral. For Weldon, that's your starting point when you want to get serious about inheritance in 1902. Weldon dies, as I said, in 1906. 2 years later, a chap called Volterek in Germany publishes a paper on Daphnia in which he compares different kinds of what we now call phenotypic plasticity in different races of Daphnia, right, growing in different ponds in the area where he was working. And he represents these graphically in what he calls a reaction norm. So the notion of the image of a reaction norm, right, showing you at a glance how phenotype varies for different genotypes as environment varies, traces back through Volterek, I would say, to Weldon. Contrast that as a starting point for thinking about inheritance to where we start our students, right, with the Punnett square. It's not often drawn to your attention that part of what makes Punnett squares teachable and accessible is that to get the right answer, the student has to think about nothing other than allelic combinations. How different a reaction norm diagram, especially multiple reaction norms in what we now call a G by E diagram, would be as a starting point. So as I say, Weldon, between 1904 and 1905, set himself to work on a book, Theory of Inheritance, which would set out his positive vision, having spent several years trying to criticize Mendelian papers and getting nowhere. For, again, reasons that I'm pleased that I think I've worked out in the book. Part of William Bateson's brilliance as the founder of a new science was his ability to generate a whole raft of exculpatory explanations, as a former teacher of mine used to call it, right? Moments when your theory and the world don't meet. Does that mean you give up on your theory? Not necessarily, because there could be all sorts of reasons, which are still good for your theory, why theory and world didn't meet there. Bateson was brilliant at that. And I think it's deeply ironic that the one-liner he's best known for is treasure your exceptions. William Bateson least treasured exceptions. Bateson was a genius at neutralizing exceptions that apparently threatened his program. So Weldon comes up with an alternative program in this book, which has lain in the archive at UCL, along with the transcript of the lecture that I just gave you a little snippet from. And I just want to boil it down to the three dicta and an upshot in inheritance. The first is that variation matters, right? We should want a science of inheritance, which is true to what we observe, not to some fictionalized, idealized version of what we observe. And Weldon thought that's true of great science. You know, some of the best science of his day, world-altering science, was science where people sweated it when there were discrepancies between what their theory said and what was in the world. An example he liked to teach with was the discovery of argon, right? The whole column of the periodic table, the noble gases, appears because Lord Rayleigh sweated the small stuff, right? When he collected nitrogen by two different methods, there was a tiny discrepancy. Lesser scientists would have thought, oh, close enough. But Rayleigh pursued it. And out of that came the noble gases. So that was a really important theme to Weldon, right? You have to be true to what you observe, and you have to remember when you've idealized. And because sometimes it can be the world that's variable in ways that might lead you on to further discovery. So variation matters. Secondly, ancestry matters. In a Mendelian experiment, once you've got your true breeding parental forms, you're excused from further curiosity about their backstories. All you need to know is that they're true breeding. Weldon thought that was a mistake. You ought to be curious about the backstories, because the backstories can ultimately throw light on what might otherwise seem to you uninteresting discrepancies between what you expected and what you observed. Again, there's enormous variability in biology. That's why we need statistics to describe it properly, but we also want properly to explain it by taking ancestry seriously and also by taking environment seriously. Weldon thought if there was one thing that was well established by the early 20th century in biology, it was that no tissue just has some properties in some absolute way, which are visible no matter what. Every tissue manifests the properties it does because of what it's interacting with. That, he thought, should be taken for granted by now. That had been beautifully established by 20 years of experimental work. What was true of tissues in general was surely going to be true of chromosomes too. And the upshot that he draws in the book is that the Mendelian picture of dominance is absolute, right? With yellowness, dominant to greenness, full stop across the board, no matter the context, is at best deeply misleading. A little gag to underscore the point, familiar to probably everyone here, but give it a go anyway. Guys walking down the street at night, sees another guy down on the ground. First guy says, "What are you doing?" Second guy says, "Oh, I dropped my keys on the other side of the street." First guy says, "Well, why are you looking over here?" Second guy says, "Well, the light's better here." Some smiles here in the room, maybe online. Right, the point being that with the Mendelian experiment, you're looking where the light is good. And that can be important sometimes. And you can learn things, but it's not necessarily where the key lies. The key is unlocking the mysteries of inheritance. So I've been working on this for some while as a historian and asking myself, yes, this debate is interesting, threw up lots of interesting ideas, but did it really matter? So what? Were we bound one way or the other to end up with something very similar to the Mendelian conception of the gene, organizing our knowledge of heredity? Maybe it was just kind of inevitable, no matter what happened between these two guys in the early 20th century. So by way of investigating that, I thought, well, I can't kind of go back and rerun their debate and let Weldon live and see what would have happened. But I can ask myself, well, what would textbooks look like now if they came out of a past in which Weldon's book had been able to have the influence that it might have had? What would those textbooks look like? They wouldn't start kids off with Mendel and his peas. Probably, if anything, you'd get a kind of reversal from what's emphasized now and what's secondary. Right? All of everything that we now know about the way that characters are modifiable in different genetic backgrounds and an environment, they're all in the textbooks, they're all in classes, but they're kind of treated as cognitive luxury items. You can learn them if you're a particularly good student, but the things that you're tested on, the things that matter, are Mendel and his peas extended. And I was emboldened in thinking we might try something different by precisely noticing that you get these points made in textbooks, just not emphasized. Our most recent encounter, but one relevant here, was in July during the big Bristol conference that George hosted. I was in Brno for the big Mendel conference there, and I went on a tour of the Mendel Museum. And what you find at the very end of the tour, kind of you've now read the policy, you get to the fine print, more or less the last thing you read in the exhibit. I'm sorry for the rubbish photo, but I was moving fast. But here's what, just so we get something about polygenic inheritance and environmental effect, just to read a little bit. What do we read? Polygenic inheritance means that a given phenotype is coded by multiple genes. This is the case for the vast majority of traits, which makes their heredity a very complicated matter. And then under environmental effect, you've no doubt noticed that if you don't water a plant with genes for tall height, those genes won't be enough to make it tall. The environment has a powerful effect on how our genes are expressed and produces even greater variability throughout populations. So what if you taught a genetics course where you reversed the priorities and you made all of that what you hoped kids would remember? And you treated Mendelian genetics as a special case, a limiting case. What happens when you dial down all the internal variability and all the external variability, so that in the simplest case, there's one difference at one locus on one chromosome. But that's not the key to everything else. That's a special case. Right, so suppose you started them off with something like the role that genes play in the condition of a heart. And you hammered home throughout, as I've said, the interaction, right, as Weldon said, that all the time, barring special cases, what we should expect is to find that there is interaction genes with genes, genes with environments, environments within the body, environments without the body. And again, when they meet Mendel, they meet him as having found important patterns, but ones that are of limited value. What would those students be like if you could teach such a course? In particular, might they be less determinist in their attitudes to genes than students coming out of a traditional course? Well, I'm happy to say that with the colleagues at Leeds, we ran the experiment. This is Annie Jameson, who was a postdoc with me on the project. She actually wrote the course, delivered it. There are all kinds of ways we could talk about in the Q&A in which, as a pilot study, there were limitations in our method, but we had two groups, right? Students taking Mendelism 101 and the students taking Weldonism 101. And what we found was that at we assessed their attitudes to genetic determinism before and after teaching. And what we found was that the students taking the Mendelian course, right, the Start with Mendel course, were as determinist about genes at the end of teaching as they were at the start, maybe a little more so. So nothing they learned in the course of the teaching had made them, on average, less determinist in their attitude to genes, whereas students taking our course had become less determinist on average. And I think in the 21st century, that's what we want, right? That's a win. It's really the more you learn about genes in bodies, the less determinist that you become about their power over us in body and mind. We published a paper in 2017, and I'm pleased to say it's gone on to become the basis for a much larger scale, vastly more rigorous, for George, I'll say, with randomized controlled trials study, funded by the National Science Foundation and co-led by Brian Donovan at BSCS and Michelle Smith at Cornell University and myself. And we're here looking much more rigorously at this question of how the structuring of a genetics curriculum affects student learning and student attitudes to genetic determinism, and also investigating potential psychological consequences and mechanism, right? Looking in particular at connections, hypothesized connections between categorical thinking, right? Yellowness, greenness, male, female, racial thinking, I'll come back to in a moment, and stereotypes, and the willingness to stereotypes or to characterize oneself under a stereotype, and what psychologists call fixed mindsets or growth mindsets, right? A fixed mindset being one in which you think that people just are the way they are, they can't change, you can't change. It goes along with a certain pessimism about the ability to challenge sexism and racism, for example. By contrast, a growth mindset is one where you suppose that things can change. You can change, other people can change. It's worth making a fuss. So we're investigating all of those links, as well as the questions I mentioned about the curriculum. And we're energized in part because of results Brian found in a study he did on the teaching of Mendelian genetics with middle school children in San Francisco. So a classic move after you've taught kids about peas is say, it's not just peas that this matters for. Let me tell you about some human diseases whose causal underpinnings look just like what I just taught you about peas. Cystic fibrosis with white Europeans, sickle cell anemia with people of Black African ancestry, Tay-Sachs with people of Jewish Ashkenazi ancestry. And what Brian found was that after those lessons, students had come away with exaggerated notions of the homogeneity of the different human races. They had come away with increased pessimism about the ability to do anything to reduce educational attainment differences between the races. They even came away less willing to socialize across different racial groups. As one of their teachers said, it was as if the kids came away from the lesson convinced that different kinds of people are different kinds of animal. So there are potentially big issues at stake in the role that we give to Mendelian patterns and processes in our teaching of genetics, and even a little acquaintance with the role of Mendelism in the history of eugenics, I think affirms one sense that there's something important to be done here. In my book, the way I put it is that the eugenic bandwagon would have rolled anyway, but Mendelism greased the wheels. Or as another colleague of mine put it, Mendelism radicalized eugenics. Now, you might think, all of that aside, isn't it important to keep things simple for students? You start simple, get them in, and then you tell them that stuff I taught you last year, it's a little more complicated than that. Well, that might be generally true, but you might think that actually with genetics it's different. And this was put, I think, very well by the psychologist Stephen Heine in his book, DNA is Not Destiny, writing actually about my experimental work. He said, there's a key difference between the complexities of the atom and the complexities of our genomes. We don't make life decisions on the basis of our understanding about how subatomic particles operate. Incorrectly believing that the atom is akin to a miniature solar system has no consequences in our daily lives. In stark contrast, incorrectly believing that genes are like switches leads us to become fatalistic and can result in increased sexism, racism, and irrational fears about our future disease risk. When it comes to our genomes, I submit that it's far better to highlight the difficulty in understanding the complex machinery of genetics than it is to give people a false sense of understanding that leads them to make potentially costly decisions in their lives. So that's my skeptical case against these three items of received wisdom. You might ask yourself, well, that's not a very fitting anniversary present for Gregor Mendel. But I don't think that's the right way to look at it, because Mendel was a brilliant man of science and a brilliant teacher. And I think the last thing that Mendel would want is for us to uncritically perpetuate what we know to be falsehoods about the past and about the present, about science. I think we owe our students better than that, and we owe Mendel better than that, right? So I think if you genuinely care about Mendel, then I think you should embrace the case I've given you today. Thank you very much.
That was great. I'm looking forward to the 600-page version. I'm just going to stop sharing so people can, so if people online have questions, I can. Kirsten's clapping. There you go. We can't hear you. But we can. If you're really successful here. Yeah. So if people online have questions, they can unmute themselves and speak. But it's left me with this is from Kirsten. My eldest son came home from school last week, and knowing my interest in the subject announced we did about Mendel with GCSE biology exams looming in a couple of months now. I'm wondering, do I sit him down and say about Mendel?
Thank you, Kirsten. Great question, and it's one I've thought about because I've been actually pretty active in working with secondary school biology teachers. They're admittedly a self-selecting group. These are secondary school biology teachers who want to enrich their teaching, who want to learn new things. And in talking with them, we talk about how, look, Punnett squares, until the revolution, which is a while off, right? Punnett squares are here to stay, and it's a disservice. The teachers do a disservice to students if they don't teach them to solve Punnett square problems correctly. So how do you do that? And yet at the same time, not reinforce this false sense that actually all you need to think about is genes really, and the rest is just kind of complexity that washes out. And my suggestion is that in the first instance, they tell students that every Punnett square should be thought of as having a sign hanging over it. And that sign says, all else being equal. So all else being equal, when we've dialed down all the variability that there might be in the environment and all the variability that there might be inside a genome and inside a body, so that there's one difference at one locus, at one chromosome, or two differences, or three, however many it is, but it's all been controlled, all that other variability has been controlled for. That's what you're going to get. And that's important. But I think that the way to come out of it without a false sense of its place in wider understanding of inheritance is to start there and to emphasize that what you're looking at in a Punnett square is a situation where all the variability has been dialed down, not at the kind of pure state of inheritance. And I'm glad you liked the joke. Thanks for letting me know.
Oh, thanks, Greg. Yeah, thanks again for a great talk. That's really helpful. I might have to have a quick chat with you about that, because I've been invited into his school in a few weeks' time to give a talk about discovery in the context of history of medicine. And I was thinking about Mendel, but I thought, oh my goodness, how's that going to go down if GCSEs are like 4 weeks away and all of a sudden I rock up, basically throw the cat amongst the pigeons. So, yeah, basically show up and say, hey, kids, now take the red pill, I'm gonna, so yeah, I might have to have a chat with you about that, but thanks again for a great talk there, Greg. Really looking forward to the book.
Thank you, Kirsten. And I say that I haven't bought, the issues here are, in some ways, they touch on issues that are fundamental to anyone that teaches. So what I found teaching teachers is that they appreciate a chance to be reflective about those moments in their science teaching when they know that what they're being asked to teach students doesn't match what their students can actually observe. And they find it deeply unsatisfying to have to say, yeah, it's all actually much more complicated than that, but gosh, we've got to get on to the next bit of the curriculum. But thank you for your question, Kirsten.
Marcia, do you want to, you can ask it or I can read it out. So Marcia's put, if Bateson had died in 1906 and Weldon lived, how might heredity have developed? One case to consider is how this might have affected T.H. Morgan's work on heredity, drawing on Reese's view of mutation?
Oh, wow. Marcia, I've never asked myself that question. That's brilliant. Thank you very much for putting it. I'm going to go away and think more about it, but on the hoof, what we see in the book, and here I have to say I drew a lot on your own outstanding work on all of this, is that by 1906, by the time that Weldon dies, Bateson has already got an extraordinary group of people working with him at Cambridge, and he's already caught the attention of a wider audience. And you see that very visibly, and I have this in my book, I show, so Weldon dies in April 1906. In July, there's the big third hybridization conference. It gets renamed the third genetics conference in the course of the conference, right? Bateson's new term gets adopted. And you just see the enormous number of people who are there, all of them, or many of them there precisely to celebrate, to extend the new Mendelian program. So I think it certainly isn't the case that Mendelism would have shriveled by 1906. If anything, right, there would have been a new martyr, for the group assembled at Cambridge to be energized by in the work that they were doing, but they were going to do that work anyway. You mentioned Morgan, and Morgan's a really, really interesting case because Morgan famously, up until that key moment, right, when the mutant fly appeared in his lab, was really skeptical about Mendelian genes. He's really rude about them. He's very funny. He quotes them, and he says, they just some new departure from what they expected comes up, and so they just invent some new factor, willy-nilly. What I point out in the book is that the mechanism of Mendelian heredity, which is the book that comes out of the Morgan lab in 1915, is a Weldonian book in that Morgan comes out of the same world as Weldon, experimental embryology. Right? He's deeply wedded to the notion that what you see in a body depends fundamentally on what's interacting with what. And I think what's so brilliant about that book, The Mechanism of Mendelian Heredity, is that it's absolutely clear that what they're reporting is what happens when they dial down all the internal variability, dial down all the external variability, and saw what they got. And they're absolutely clear about how under other conditions, other genetic backgrounds, other environments, all bets are off. So I think Morgan's a really, really interesting case. And in a kind of cheeky way, my book ends with 3 postscripts. And one of them is the addition of the Dictionary of Scientific Biography from the counterfactual past in which Weldon lived. So I give him an extra 2 years of life in my counterfactual fantasy. But I point out that in that counterfactual world, the book that Morgan publishes is called The Mechanism of Weldonian Heredity. That's how close I think they were intellectually. And it's really important to emphasize that in the book that Weldon was working on when he died, Mendelian patterns are given their due. In Weldon's view, in the 19th century, there were really 2 class acts on heredity. One was Mendel and the other was Galton. And these were the 2 men who'd done most, both to produce statistical descriptions of the phenomena and then to explain the physical basis of the patterns that they'd identified. And so the choice between them is between 2 different conceptions of dominance. There is, with Mendel, the absolute conception in which greenness is just recessive, yellowness is just dominant. Whereas with Galton, and we're not at all used to thinking about Galton this way, with Galton you get a relative conception, where what's dominant or recessive can be different depending on context. But Marcia, thank you so much for the question.
So, Kirsten's here, so he can correct me because but I'm half German, but I can't read German that well. But I mean, in Mendel's paper, he's very clear about the fact that most of he looked at lots and lots of traits, and most of them basically blended.
Yeah.
And so so it was a methodological decision, or it could be seen as a methodological decision to take the ones that he could actually study through the fact that you could actually see it in terms of the trait.
Yeah.
But that he could very well have thought the processes he discussed, which became known as segregation, independent assortment, were still underlying other forms of transmission. It's just you could see this with the dominant traits, and in some places there were we all know Mendel's two laws in the UK generally, but in other places, also in Germany, you get the third law.
Yeah. Law of dominance. Yeah.
But that wasn't a law. You don't see that as the law. The key thing that he put forward is that essentially what we'd now say is that inheritance. Genetic inheritance, not trait inheritance, because traits are influenced by many other things. The genetic inheritance underlying that is a Markov process. So, which is what those two laws say. I.e., it doesn't matter what came before the state before you, but the things that come before you, which is the ancestry that Weldon talks about. In terms of what you actually get genetically, it doesn't matter, because if it hasn't got to your parents, it can't get to you. And that's what the two laws say, and that's what we now know from massive sequence data, etc., is the case. And so that if those were taken, I think Weinberg is the first person I saw who in 1908 in his paper, which is generally quoted for the Hardy-Weinberg equilibrium, but Weinberg thought that was trivial. He's talking about many other things in that, and he basically presents it as that this is what the Mendelian inheritance is that it's Markov wasn't around then, so you couldn't call it a Markov process, but he describes it in that way, that basically it doesn't matter what's gone before, and that's the genius of, or if you like, the thing which transformed understanding, and that's carried on. That's Bristol was known for Mendelian randomization.
Yeah. Of course, right, and so. I'm still in the foothills in my understanding of Mendelian randomization, but if the one important element in that very rich comment was the notion that what we esteem, one thing we really esteem about Mendel is this idea that it's random whether the zygote gets this allele or that allele.
Yeah.
Right. So is that in the paper? Absolutely.
Yeah.
Is it totally distinctive of Mendel? No one else thought that? Well, right. So what you find is that actually, so there were not a lot of people who became thoughtful about these kinds of processes, but one of them, independently of Gregor Mendel, was Francis Galton. Another was Charles Darwin. And if you were to ask Darwin and Mendel, so what is it that leads for Darwin this particular gemule? And Galton used that vocabulary for a while. Then he called it particle. To end up in why does that gemule end up in the gamete rather than that gemule?
Yeah.
It's not going to do in any kind of important sense with the past, right? Which is where your talk about Markov comes in. It's going to be partly a matter of internal conditions, right? And what we call randomization, of course, at a certain level, you ask, well, is it that we can model these things as if they're random, or is it kind of genuinely causally random? And Galton and Darwin are very thoughtful about how, to a certain extent, what we call chance means causation that we can't track. At a certain point, it kind of doesn't matter. But the idea that it's effectively random, what ends up in a particular gamete, that's not distinctive of Mendel. And my own sense is that from 1900, that's probably the view that pretty much everyone else would have had. And so, yeah, I think Mendelian, to call that kind of randomization Mendelian, it's an honorific, but I don't think the label does a whole lot more than that.
But I mean, the actual, the independent assortment is sort of true, outside of being on the same chromosome. I mean, it was getting a step further than Darwin or Goldman. There's
a fantastic paper to be written by someone, maybe by me one day, called Mendel's Luck, because it's a theme that comes up a lot in writing about Mendel. Gosh, how lucky he was to choose peas. Gosh, how lucky he was to choose these characters, which turn out to be on different chromosomes, and so
two of them were on the same chromosome, but one of two are a reasonable distance, and two of them were actually close and should have shown.
So even more, right? How lucky he got, right? Yeah, and so that's a tricky paper for someone to write one day. But again, if you ask yourself, what is it that assorts independently? You said it. It's chromosomes, right? And so the independent assortment of chromosomes was something that would have been worked out, right? And it would have been worked out by people thinking about chromosomes. And it's an interesting feature of the history of biology that a guy who, for his own purposes, not least so that his weird mathematics where he, unlike our textbooks, right? When Mendel gets to dihybrid and trihybrid crosses, he multiplies A plus 2AA plus B by B plus 2BB, and he works it. He shows how it all maps onto his piece in a really complex way. But for that maths to make any sense, he needs independent assortment to be true. And there's something more to be said about quite how all of that ends up working out the way that it does. But I think that the fundamental point about random distribution is I think less distinctively Mendelian than you've been led to believe.
Well, I'll follow that. The last point that you finished on eugenics, Weldon's closest colleague and co-thinker Carl Pearson, in 1897, wrote about how Britain was doing itself down because of the sedulous nursery of inferior kids, that you actually wanted them to die in infancy. Yeah. So he was someone who shared Weldon's view shared Weldon's view and was anti-Mendelian in the same way, but came up with the most appalling form of eugenics you can imagine. And Pearson had been in charge anyway. So I'm not convinced by the notion that the eugenics was strengthened by Mendelism. It's difficult to get beyond Pearson for the level of vicious eugenics.
Pearson is a piece of work, but we all have friends, right? And we have allies even that we disagree with, and maybe even don't get us. And when it comes to everything I emphasized in the lecture today about how profoundly Weldon took the fact that organisms develop, and that development always takes place in the context, and we can't ever forget that. Pearson didn't get that. And Weldon really tried to rub his face in it. And Pearson just was a hereditarian to his fingertips, which, by the way, made him, his attitude to Mendelism is complex, but in the period that I'm talking about, Pearson was leading the way and saying, why are we fighting? Surely we can all see that these two theories are parts of one greater whole. So Pearson was not Weldon. And this comes through, I hope, very firmly in the book. So on the one side, Pearson just didn't take development environment seriously, right, at all, despite Weldon really trying to make him do so. And Weldon was pretty snippy sometimes about Pearson being a maths major who was now heading into biology, but he didn't have a biologist sense for what was real. And so, he's often quite funny and cutting about the ways in which the problem with mathematicians getting biological is that they're just formula-led. And it takes a biologist to know where the formulas are helping and where they're just leading you astray. Yeah, so they were friends. They were really close friends, but they weren't singing off the same hymn sheet.
I wanted to First of all, it's brilliant, and I love the way you brought back history and these two in particular, three, I guess, people to illustrate it. And I'm going to read your book because you sold me on it. At the same time, I'm struck by how much we identify different people in history and we reinforce them and minimize others.
Yes.
So clearly it wasn't just because Weldon died that genetic determinism has been bolstered and bolstered and bolstered. And if these kids that you're talking about teaching, which I love the way you're talking about it, NIH's portfolio is going to change dramatically, which would be all for the better, because it puts way too much towards genetic determinism in various forms. So who benefits? Why did we continue to reinforce that concept of determinism? And why do we still do it today?
I appreciate everything you said in your comment, and just to pick up on a few things. To start with the kind of pessimism, like, well, you know, surely there's from so many different directions. Determinism is reinforced. You know, why should we think that this one difference in the past, however important, however good it might have been, would have made a difference? And there are several ways into this, but just to touch on one that I've been thinking about recently. We've just lived through the COVID period. Most people have no trouble at all with the notion that the same microbe in one person can bring on lethal illness, in another person, leave them absolutely fine. Nobody struggles with that, right? And in other areas of biology too, you can get elementary school kids to understand Darwinian adaptation, right? So they understand that actually, a woodpecker isn't universally well adapted. A woodpecker is well adapted to the environment in which its ancestors evolved. So both in our biology teaching, but even like when I think of COVID, general understanding, there's no general problem with people thinking about variability in context. The problem is kind of isolated. It's specifically with genetics. And in the book, I investigate various explanations that historians have favored for why genetic thinking took over the shop in the way that it did. And there are some who have given quasi-Marxist readings, right, which say that unequal societies need some story to tell people who are lower down in the pecking order about how their place in things is foreordained. It's determined. And in the past, that came from the church. And in our less religious days, it comes from science. And so our societies remain stable. Our capitalist unequal societies remain stable because the people to whom we give authority, right, the scientists, are there to say, it's just luck of the draw, right? Some people end up at the top, some end up at the bottom. And you know who emphasized this theme? William Bateson. I draw attention to this a lot in the book. Bateson, in the First World War, went to give popular talks on Mendelism to soldiers. And a Scottish soldier, after he listened to him, said, "What you're telling us is scientific Calvinism." And Bateson says, yeah, that's about right. In fact, Bateson wanted to use that as the title of his popular book, the popular book he never wrote. So I do think that, the case of genetics is, so I go on to criticize. I think there's something to be said, of course, for the story, the Marxist story, but I go on to criticize these larger kind of superstructural explanations and to suggest that, no, actually the explanation we want is to do with what happened in the early 20th century, and that it wasn't inevitable that we would end up teaching and thinking the way that we do about genes. And one way of getting clear on that is seeing that actually there was this alternative that was well articulated. It was as prestigious, in its institutional bases as the Mendelian alternative. And I think it would have worked out to have similar kinds of prestige within applied work in heredity, in agriculture and in society. I think it's important one reason I think that the teaching experiment is important is because it is easy to just be pessimistic about all of this. Whereas I think that what the teaching experiment shows is that, the classroom is potentially potent, and the right lessons at the right time can produce students who at least have a kind of intellectual prophylactic when they get out there and find that a 23andMe ad is trying to convince them that actually there's just this gene and you've got it or you don't. As Heine says in his remark, there's just some switch and switch, boom, you're yellow, switch, boom, you're green. It'd be great to produce kids who get out into the world and are just able to put that at arm's length.
So I think we all agree with that. We all go against the 23andMe ad. It's like a kind of back again, just a brilliant talk. And we were going to carry on talking in the pub, so if anyone wants to.
Thanks, thanks Kirsten, thanks Ida, thanks Martin.
Just to say to Kirsten about the Daphne experiment, that'd be a great idea if it got in the GCSE biology textbooks, but it should also what should get in is W.E. Agar, who a few years after Weldon did Daphne experiments and thought he had demonstrated the inheritance of acquired characteristics, and he did these. But then, as a brilliant scientist, he then went on and redid that, standardized things and etc. Showed that it was basically Markovian, and then spent 25 years of his life trying to repeat people's experiments from these people like William one of these McDougall,
yeah.
who was the Markians were also eugenicists as much as the non-Markians.
As I say, it's not you can't say Mendelism and eugenics 101, but there's a story to be told.
We should have Agar's Daphne experiments and his later recanting of his work on that as well.
I mean, a great question. Thank you, Marcia, for the question. Are the questions preserved in the online?
Yeah.
Great.
Yeah.
I owe you answers, Marcia and others. Thank you very much, and thanks to those who came today.
Cheers. Thanks so much. That was brilliant. That was.
It's my pleasure.
Lovely. So, if anyone would like to come to the Victoria.